You mentioned cost. There was a question that came up earlier about cost per sample, and you mentioned cost effectiveness as a part of this, the value of eDNA and this technique. Do you have can you answer the question about how much it costs versus other sampling methodologies?
I think the answer can be pretty different, like depending on the question you're asking and what you're looking at, what you're including in your costs. But so if you just compare the costs, for instance, of what we're doing for tiger detection, like just the occupancy survey, so going and relying on the detection of the signs to be able to assess the unbiased presence of the species in a particular area. Usually it's a 15 or 16 by 16 square kilometer. People need to walk between 40 and 100 kilometers to get good enough detection probability of the species when it's present. So the collection is on tiger and other mammal species, and that costs, I think, I would say between $5,000 and $10,000. So if you really if the sampling of water in a river can really be can demonstrate its accuracy and efficiency, and if you need just one sample or two samples to cover the same area, then you could probably do the same sampling for less than $1,000.
Wow, that's crazy.
So cutting the cost by 5 or 10.
Do you know how much the cost is per sample?
Yeah, it might be different, but it's around like $300, $400.
Okay, cool. I'm going to invite the other two speakers into the discussion. Inka, you had a question that you were discussing with Kat. Have you had a full answer, or do you want me to throw it to the group? Ooh.
By all means, if anyone more experienced with this stuff wants to help me out.
Do you want to, Kat, can you paraphrase what she's asking?
Yes, so let me just...
Do you want to throw your video on?
Mm-hmm. Yeah. It was about it's sort of starting to touch on what state DNA is in, and whether how degraded it is. And I think this is something where there has been an evolution in our understanding over the last few years, because initially it was thought that sort of all of the eDNA that we're picking up is basically extracellular. So it's DNA where it's come out of the cells and out of the mitochondria and is just floating around as sort of free-form DNA, and therefore will be very broken down and degraded and only available in sort of very short sections. And I think that over time, thinking has moved on a bit from that, and that actually a lot of the DNA that we're picking up in the environment is still either in the form of cells or of organelles, so potentially sort of mitochondria that are floating around, or even tiny bits of tissue or scat or whatever. So it's not all extracellular DNA, and there have been some nice studies that have looked at size fractionation. So if you pass water through different sizes of filters, on what sort of size of filter do you pick up most of the genetic signal. And there has been one study in particular where they managed to get full mitochondrial genomes to be amplified from water samples. That is something that I haven't done personally. I don't know if anybody else has done this or tried it. It's not as sensitive. We still get, I think, the best sensitivity from using short markers. So it tends to be a trade-off between shorter markers are more sensitive, so you pick things up more readily. But the disadvantage of them is that it can be more difficult to separate species because you've got, in a way, sort of less information to separate them on. If you go to slightly longer markers, then yeah, you have the opposite issue. So it can become slightly less sensitive, but you can get better species level resolution. So there's always a bit of a trade-off there. There's quite a lot of work, I think, about to start on really understanding more exactly what form eDNA is in, and that'll be useful for everyone because it feeds into how we handle it and how we extract it most effectively. So an area of active research.
Did any of the other speakers want to comment there?
Just to add a few things. Actually, DNA breakdown.
Alice, do you want to just turn on your video?
Yeah, sorry.
There you go.
Actually, DNA breakdown in the environment. So you can have metagenomes, but you have a higher probability to have a shorter fragment to find rare species because shorter fragments are more present in the environment.
Hey Fred, do you have a mic? Oh, you don't. Okay. Fred's thrown in a question that I have written down as one I'm quite interested in to put to the group. So it came through that one of the things that seems quite important is species databases and sharing of data and sharing of information. And it kind of, that was a thread throughout all of the talks. Fred's asking, would like to find out more about the databases and ask, are they open source or are they part of companies' intellectual property, or what's the process for how shared are they and how accessible, and what are the barriers for sharing if it's not there already?
We use GenBank for database, and we also construct local database. And when the data are published, the environmental data, for example, for French Guiana, the fish database is implemented in GenBank. So it depends. This is published. Also the sequence are published. It's a private. It's still with the clients.
Kat?
Yeah, very similar answer from me. So the starting point for us as well is GenBank. The GenBank is an interesting one because there's a lot of error in it. It's sort of a universal dumping ground for sequence information, and so there's a lot of people who. My favorite example is people who think they've sequenced their study organism and they've actually sequenced themselves. And there's lots of random human sequences messing everything up. Yeah, it's uncurated. So I think I would say that we do quite a bit of sort of curation of GenBank internally so that we have a higher confidence database to work from. And we also do sort of supplement things with local reference databases where necessary. Yeah, generally those end up being shared, and I think that there's a real sort of movement towards open access for particularly sort of species records, and I think that's really important from a conservation perspective that those are accessible. So yeah, combined approach, but generally it's pretty open.
Arnaud and Robin, do you want to throw in a conservation perspective?
Yeah, no, this is definitely one of the main problem we had, and in the study that we conducted in British Columbia, we also had to complete the reference database with like species that were either not available or they were available but from a long distance, so they were not matching properly. One of the very difficult thing is when you, because yeah, you want to be conservative and keep only the species that match at like 100% or very high probability of very high confidence. But in places I've had this problem in Myanmar, for instance, where there was this deer species that was very, very abundant, but it didn't show in the data because the reference was too far away, and it would not be in the results So it decreases the confidence of people in the method when they don't see one of the species that is most abundant in their area. So it's tricky to communicate about the result and make people understand that the reason why sometimes things happen. It's like the dinosaur species or bird species that are from another continent. It's it's difficult to manage, and it really needs a translator at the moment, and also working on the database. It's a requirement, and I don't know what other speakers know about it, but those portable devices that allow to do that in the field, I've heard about Minion, and I think there are other options out there, and I would be very interested in knowing if that could be a good option for the future, because if you like provided those places with the possibility to work themselves on the reference database, that would be a really big advantage.
Kat or Alice, have you had experience with any of the portable devices?
No, well, only sort of very much on the periphery.
Why is that? Is it just because they're still, because we've got a couple, I know we've got at least 1 or 2 members who are participants who are using them. Why is that? Is it because they're too, is it just they're not collecting the right sort of data, or is it just they're too unreliable?
Yeah, it's a combination. I think they're often talked about in a way that makes them sound much easier to use than they really are. So the reality at the moment is that it's not a matter of just taking a water sample and dropping it onto the sequencer and it sequences it. You've pretty much still got to have a DNA extraction lab, and you still need to do the PCR generally if you're going to get, if you're going to get representative data that's similar to what you would get on the other platforms. And so you need to have those sorts of capabilities already. And then it's still relatively expensive for the amount of sequencing output that you get compared to the Illumina platforms that we use. And for us, the focus has always been on quality control, and so that's much easier to do in the lab environment. I do think they have a place. I think that they will sort of probably, particularly actually for barcoding and for generating reference databases, I think that they can be super useful. I think in places in the world where you don't have the larger sequences, I think working with the MinION platform within a lab setting in those places is probably a really good, has got a lot of potential. But for us, it's not a tool at the moment that's ready for sort of large scale application in a commercial sense.
Interesting. Alice?
It's more or less the same for us. We know the technology, we didn't test it yet, but for what we know is that is made it for very long fragments, and this is where the technology worked well. And because we are interested in small fragments to increase the detection of rare species, that means that it's not, sorry, it's better now?
No. My expression was just because I had an aha moment more than anything.
So the Minion is very good for long fragment sequencing. So it's better suitable for entire genomes or metagenomes. And because we are focusing with environmental DNA on small fragments, the technology is not made for us for the moment.
Interesting.
So I'm jumping in because that's the reason, that's the function I see with this. So for a place like Myanmar, when most of the species are unknown, but the species have been collected, or maybe most of them are on the meat markets, and the tissues, they're very easily available. So that's a possibility for people living in the place to go collect tissue samples and sequence the 12S or whatever gene we're using, or the mitochondrial gene. So we do improve the reference database. It's not to analyze directly the water, it's really to improve the reference database. So when we analyze the water samples or whatever other sample we have, we do have a match in a better database. So the question is, can we use Minion in these places to improve the database without having all the problem of shipping samples to the lab?
Interesting. Okay, cool. I just wanted to brings back to a question that Stephen was asking about, because you've brought the question back to the reference databases. Stephen was wondering, are there any efforts to connect the data in GenBank to other databases like the Global Biodiversity Information Facility or MoveBank, or do you know if there's any, or where the efforts are to link all of these disparate data sets up? Or
I was just writing a reply to that, so I'll jump in. I've spoken to GBIF a fair amount. They're really interested in integrating this kind of data into their platform. It needs some adaptation of their platform because they need to be able to record the right kind of metadata to go with DNA-based detections. So people will need to know, you know, what genes were used, how it was identified, and that kind of thing. So it's not as simple as just I think it would lead to confusion if records were just uploaded sort of straight away. But there's definitely a lot of potential to integrate with I think particularly with GBIF. It just needs a bit of joint effort to set it up. Process of it.
Great. Excellent. I'm aware of the time, and I just wanted to throw a question that bring this discussion to looking towards the future. And I think, I mean, out of all webinars, this one I felt the most out of my depth, because it's such a new tool, and compared to camera trapping or drones or whatever, there seems to be so much opportunity and not that much information. So I mean, I've learned a heap from just listening to these presentations. I wonder, where do you see it? It seems like it's there in terms of marine environments, aquatic environments, but where do you see the future and like it going in the next 2 years, 5 years, 10 years, and applications and effectiveness? I should direct it to someone. Alice, do you want to take this one first?
I will try to answer. So, actually, environmental DNA is a new topic right now, but looking at the publication that come out every year, you can see that they're just growing exponentially. So they really become a tool that has proven to be cost-effective, non-invasive, and so they can you can do your survey using environmental DNA quite easily, and you can do in the parallel with other surveys. So actually in the next future, more and more study will come out and demonstrate that it work on all kinds of environment, marine, terrestrial, or and there is also some study they use air samples to describe the biodiversity.
Yeah, right. Robin, I haven't heard from you yet. Where do you see the interesting paths it's taking?
I mean, this what I'm going to say is probably more on the 10-year horizon than you know, more quickly than that, but because there's obviously still a lot of the technical aspects that need to be resolved that we've discussed about here. But I was kind of struck by something Kat said in her presentation, where she had you know, citizen volunteers collecting information, and you know, we've seen the way that things like eBird has really revolutionized and given us fascinating new insights on the distribution of birds around the world. So, you know, wouldn't it be cool somewhere down the line where you've got a whole bunch of citizen scientists collecting water samples or soil samples and having an app that collects that information. Obviously it's going to be a little bit more complicated than the eBird situation, but I could envision somewhere down the line where this technique combined with citizen science could really lead to some pretty amazing breakthroughs in our understanding of the distribution of biodiversity.
I mean, that's like, there's a couple of steps before, like the ubiquitous eBird. Like, I'm just thinking of adventure scientists who like a kind of the halfway point for citizen scientists putting out camera traps and we all enjoy that. Okay, Julie, do you have a mic? Do you want to jump in?
Yes, I do. Hi, everybody. So I am really curious about the use of citizen science to collect the eDNA samples and just hear about some of the pros and cons. I know some of you are using citizen science or individuals. So what are the pros and cons, and how do you control for contamination or standardize the filtration process?
May I answer this question?
Of course.
So what we did, actually, we did a little training for all the people that go on the sampling to they're aware about the fact that they can contaminate or cross-contaminate the samples, or they can bring pathogen between the different sampling size if they are not strict in the protocols. And after we have a single-use equipment to do the filtering, and also we have encapsulated filters, so there is less contamination risk. So, and as we show in the Greek-crested newts sample, all the 200 and nearly 40 ponds were collected by volunteers. They were just people like me, like everybody else. So.
Rob, do you want to throw your question to Arnaud?
Hi, everyone. Thank you very much for a mind-blowing set of talks. I just had a question about I guess the limits of this. Like, I was really fascinated by the idea that you might be able to get DNA from a paw print, for example, and even though it might not be quite there yet, is there going to reach a point where it doesn't matter how good the technology gets, the DNA just won't be enough, or is it just a matter of the technology catching up?
I'm afraid I don't have an answer to that. It's just and it was very, very, yeah, it was a very, very small test that we did. Like the way I sampled in the mud for the Sumatran rhino was just one cotton swab by footprint, one cotton swab and one footprint, and we repeated that 10 times in 10 different footprints, and 30% of them had enough DNAs to show a signal in the in the sequencing process. So, and I did that one swab per one footprints to have an idea of the number of footprints that we would need to swab with the same cotton to make sure that we would have enough DNA on the sample. But yeah, it's just one time, right? So and we did not quantify, and I don't know if there's a way to quantify, or there's probably a way to quantify the DNA on your sample, but we haven't done it. Alice, were you were in charge of this analysis? Have you quantified the DNA that we found there?
I don't remember if we quantified the DNA, but for each experiment what we done, we calculated the limit of detection of the marker. So we know that below a certain limit it's not possible to detect it. So this is made in the same way of the analysis. So more or less depends on the type of marker. You can have more or less better detection than other markers.
So I'm conscious of the time, and we have the outstanding, I just wanted to ask Arnaud, did you have an opinion, comment, and then I'll put it to Kat, on the next 3, 5, 10 years of where you see this going?
You're asking me the question?
Yeah, so I wanted to, yeah.
I'm sometimes some colleagues say I'm overconfident, but I really see the potential, especially for the terrestrial mammal distribution. Well, because that's the thing that seems more questionable, is how do you like collect water and are able to tell all the biodiversity that is even like, that hasn't touched the water. So, and it's very complex, but I'm interested first in very basic, a question is just the detection probability of the different species, and being able to do the same kind of thing that we do with traditional science surveys, but with a better accuracy and at a much better cost. So I think if in the 2-3 years that are coming, we can really show that this is efficient, that could be a really, really big breakthrough. Because it means if we know how much water do we need to detect a rare species like the Sumatran rhino or like the tiger, we can really simplify the way we monitor these species and their prey. So that's pretty much where we're going at the moment.
Wonderful, thank you. And Kat, do you want to wrap us up?
Yeah, cool, thanks. So I think I agree with everybody about the future perspectives, although I think the citizen science is much closer than sort of 10 years away. I think we're pretty close to actually having that as something that can be very real very soon. I would say for me, the thing that I think is most exciting in the next few years is the opportunity to really think again about how we measure biodiversity. Generally, I think for a long time we've got used to the fact that it's very difficult to get a lot of direct biodiversity data, and so there's been a lot of use of proxies and indicators and things, and now we're getting to a place where we can generate biodiversity data at really quite unprecedented scales, and there's a chance to think about how we use that. I think that you've got your biodiversity as sort of charismatic fauna that everybody knows and cares about, but actually most biodiversity is small stuff. It's uncharismatic, invertebrates and things that are actually driving ecosystem function and resilience, and all of those things can also be captured and sequenced with these methods. And I think biodiversity is a big data problem. You've got millions of species responding to thousands of variables, but and there is signal in it, and I think we can start using some of the sort of big data tools that are used in other areas of life that can drive simple visualizations and metrics and scales and indicators of change, but driven by these sort of big complex data sets. And the block to that has always been the inability to take a sample of nature and turn it into data, to be able to put these sort of analytical tools on top of. And I think that DNA is part of a suite of tools, including bioacoustics and remote sensing and things which are allowing us now to do that and to generate this kind of data. And I just think it's really exciting to think about what we might be able to do with it, to be able to really monitor and make evidence-based decisions for environmental management.
What a fantastic wrap up. Thank you so much, Kat, and thank you to all our speakers. It's been a fascinating session, and sorry, I keep getting distracted by the chat. Lots of valuable information. We'll write up our notes from this session and include all of the links and papers and comments that have been shared in the chat and in the talks. Talia's just dropped in a link to the discussion thread on Wild Labs. We do have an eDNA group on Wild Labs, so if you're interested in this topic, it feels like a lot of you, especially in the introductions, are working in this space, and it would be valuable. It's a small world, it seems, but if you want to connect in the eDNA group, it's there for you. Thanks, Talia. We have one more meetup in this series. It's going to be about acoustic monitoring early next year, so keep an eye out for that. We'll have the recording and notes posted in the next week or so through the same link you used to find out about this talk. And wonderful, and thanks as always to Talia, who makes this happen behind the scenes. And we'll see you all very soon. Oh, and have a wonderful festive break if we don't see you before then. Yes, thanks everyone. Speak to you soon.